Thank you, Gary, for the opportunity to present on DNHG Members Night, December 2024. Let's begin. So this is a presentation on my personal journey on arthropod identification. I've structured the presentation into the following four parts. First, personal motivations for choosing arthropod identification and why one might be motivated to observe arthropods more closely. Second, identification basics, some do's and don'ts which I've learned the hard way in the field. Third, some case studies on butterfly, moth, and odonate identification, which are relatively easy to attempt identification for on the basis of visual cues alone. Here we'll go through some case studies, and these are from my own photographs from the UAE. Lastly, resources for identification which have helped me thus far. So this is the structure considering the time limit of 20 minutes. So let's begin. Attempting arthropod identification, personal motivations. So again, the following 4 points motivated me to pick arthropod identification. First, species diversity. As many of you may have observed in the field, there is amazing species diversity in arthropods, a lot of which are not visible to the naked eye. More importantly, it is a means to explore nature much more consciously. A lot of you may be into mammals and birds, and we don't tend to look at small things closely, but they play a critical role in the ecosystem, and science itself has benefited greatly from learning about this. I don't know how many of you may be aware, but the Novavax COVID-19 vaccine was developed in the cells of the army moth larva, and specifically the species Podoptera frugiverda, which is considered a so-called pest. It's only if we attempt to know a little bit more about insects can we learn how critical they are to the ecosystem, theirs and ours. I'll expand on this in the forthcoming slides as well. Second is the ease of attempting basic arthropod identification. And I emphasize basic here because, of course, we are not attempting species level identification, but at most to the tribe or the genus level, based on visual cues. I'll illustrate this via case studies. Third, meaningful participation in citizen science initiatives in India. This, of course, is primarily on account of my work in India. In Delhi, especially, a lot of ecosystems have been polluted due to development activities and, in general, human interference. But there is a dedicated center for restoring degraded ecosystems, which has done a fantastic job of attempting to restore these, at least in part, over the past 2 decades. One of the bioindicators used is the presence-absence of specific arthropods. And I've had a chance to participate in annual counts, which are done for odonates, moths, butterflies, and so on. You will see firsthand what is in textbooks actually playing out in the field as these ecosystems are restored. Lastly, the chance to practice macro photography. So if you're specifically into macro photography, arthropod identification will help you to pursue this vocation with rigor. So this is broadly what motivated me. Coming back to the point on species diversity, what I've cited here is a 2017 article from Nature, which examines taxonomic bias in biodiversity observations and societal preferences relative to species diversity on the basis of data sourced from the Bing Internet Search Engine. The authors have scaled the axis. As you can see, the x-axis is not linear. Rather, it's a specific transformation called the inverse binomial sign transformation, which allows them to represent the abundance in occurrences of reported observations for specific groups on the basis of species diversity. You can see birds right at the top and insects right at the bottom. And this difference couldn't be more stark because it's not linear, it's several orders of magnitude higher for birds relative to insects. The truth is we don't know many of the insect species around us, and they're getting extinct faster than we choose to acknowledge. So again, coming back to the point on bioindicators, arthropods served as reliable bioindicators. So this is a table summarized from a 2023 paper on insects as bioindicators for various ecosystems. 6 groups of insects have been represented here: butterflies, moths, dragonflies and damselflies, caddisflies, beetles, crane flies, uh, hoverflies and mosquitoes, and ants, bees, and wasps. The table shows that the health of the ecosystem is directly proportional to the presence-absence of specific insects. And this is a compelling reason to pursue the identification of arthropods, because it is something that may have policy implications for those who want to develop quote-unquote smart cities or green cities, and may benefit from having a ready-made gauge to measure their effects and initiatives. The reference paper calls out many more insect groups so as to what they track as bioindicators, and you would benefit from reading it separately. So let's proceed to the next part, which is some identification do's and don'ts, which I've learned the hard way in the field. Point one, capture photographic or videographic evidence. And I've structured this entire slide into bullets, and while each of these can be a separate lecture by themselves, I'll just brief you on each of these points. So first, of course, capturing photographic and videographic evidence. For the majority of my lifetime observing nature, I didn't have a camera. And this is something which certainly helps in arthropod identification. I would recommend a DSLR camera with a lens of focal length around 70 to 300 mm should be adequate. And the main reason being that a lot of the specificities for which help to identify arthropods may not be visible readily to the naked eye. And the second thing is that this focal length is adequate because many arthropods do not permit closer approach, especially butterflies, dragonflies. Maybe some spiders may be kind enough, but most don't. So one can take photographs from a safe distance. Second, aim to capture lateral, dorsal, and frontal views. And this is again one point I wish to emphasize, because even for experts who want to help out, scan data will make their job even more difficult. And for anybody attempting identification, it will help to have as many angles as possible and in good light. So try to capture more angles of the same specimen. Third, explore in a variety of habitat. To put it simply, there is a science, there is an art and a craft to arthropod identification, and field exploration helps to hone the craft. One gets a little bit more knowledgeable about where one can expect specific diversity of species on the basis of exploring in various habitats. So I would encourage this. The next point being jotting down observation data plus metadata. Especially photographic evidence is just one part of data, but it doesn't really help in its entirety with the endeavor of species identification. Reason being that there is a lot more that is not captured in the photo. For example, what was the behavior of that particular insect? What time of year was it observed? The habitat in which it was observed, the geography in which it was observed. Next, did you see any species nearby, native species or those which have been reported in the same geography? So all of these are metadata pertaining to that particular observation, which can help to make the job of identification a little bit easier. The next point being verification via internet-based resources. These days, unlike earlier days, we do have access to digital books and scanned copies of specimens from various museums, natural history museums worldwide. And this is not a resource which was available readily earlier. This is in addition to physical books which may be available for specific types of arthropods. I'll go into this particular point in greater depth in the resources slide. But this is certainly something which helps to corroborate what we are seeing out there. The next point being limiting to the ID to family genus level or tribe level. The reason being that, see, it's like with on basis of visual cues alone, it is many times a very almost impossible endeavor to get the species right. Full-time researchers have access to labs and processes such as molecular phylogeny or examination of the genitalia, which allow them to identify species. So it's perfectly fine if you get to the family, tribe, or genus level. There is no need to go further, and you're doing really great if you're able to get thus far. Lastly, seek experts' opinions. Uh, so if you've done points 1 to 4, it makes approaching experts much easier. Most of the experts who are working in the space of arthropod identification are overburdened due to the number of projects, due to funding, and a variety of reasons. So it will help to present a rich data set when you're seeking an opinion, along with the rationale as to why you're placing a particular insect in a particular taxonomy. Coming to don'ts, relying excessively on reverse image search. especially with platforms such as Google and iNaturalist, they are trained on available data. In geography such as the UAE and Oman, my observation is that especially for arthropods, there may be scant reported observations. Consequently, the reliability of what a reverse image search throws up is dubious. So rather than trust but verify, the dictum is just verify, specifically verify these. Many times they can throw one off completely and suggest a species which is not even observed on the same continent. And again, this is something from personal experience. So don't rely excessively on reverse image search. Second, don't attempt taxonomic identification at the species level. As I mentioned before, there are specific techniques available to research labs which may not be available to lay observers which are relying on visual cues alone. And also the principle of safety first here, because an incorrect ID takes almost double the effort to correct for, and it can lead even experts and researchers on the wrong track. I'll illustrate this with case studies as well. The third point being reporting ID for non-native or new species. So there's a temptation to, especially when you go to a geography where there aren't that many reported observations, to check and report for potential new species. But it's worth checking. One, is the observation a known native or introduced species? Sometimes these are reported in the news and are maybe under subject to existing research efforts as well. Second, if it looks similar to known species, which indicates the potential for hybridization or integration. And here specifically the phenomenon where, because sometimes individuals from multiple species may be in the same area during mating season and were not able to find a mate from their own species, tend to hybridize with members from the similar species. So what happens is you might find an individual which exhibits characteristics of both, but is not a new species. It is just a symptom of hybridization or introgression. And lastly, any other evidence to expect the species in the area at that time. And again, that comes down to metadata, which will help to make the observation point much richer. So these are some of the do's and don'ts. And we'll go through this process again when we are visiting the case studies in the next slides. Now, coming to the case studies, I have limited the observations to Lepidoptera, odonates. Why Lepidoptera being butterflies and moths, and odonates is specifically on a case study of a dragonfly. Why? One, relative ease of visual identification, and two, no requirement of physical specimen and genitalia dissection, at least for at least genus level identification. The indicative methodology for identification is the same followed for all of them. Attempting to ID the tribe or the genus via iNaturalist and personal experience. Second, consulting text and online resources. Third, consulting photographic records, illustrations, and digitized specimen collections via online resources such as Biodiversity Library and UAE Butterflies. And lastly, input from experts and corroboration for identification. So let's come to the first case study, which is on butterfly identification. So on the left-hand side, you can see an illustration from butterflies of the UAE in Northern Oman by our own Gary Fulner and Benny Shubas and others. So this illustrates the scientific names for the forewing and the hindwing of butterflies, which again enables one to interpret text easier in an easier manner. And on the right-hand side, you can see an example of a salmon Arab, Colotis calais. So this is a female, which was seen at Mushrif Park in October 2024. So to follow the methodology which was explained in the previous slide, when was this seen? October 2024, where in the desert Ghaf habitat, in the dry scrub, which is basking. The visual cue specifically here being that it is larger and less colorful than the small salmon Arab, which I've seen from personal experience in Delhi, in India. And input from literature and experts indicates that males are less colorful than females. And the Afro-Asian Colotis calais was formerly considered a subspecies of the Indian Colotis amata, but have now been separated via molecular phylogeny. With this, we can reliably say that this one is a female Colotis calais. Coming to moths. So again, from on the left-hand side, you can see illustration of the body parts of moths from the recently released Moths of India, which is an authoritative text on the list of observed moths in India. And on the right-hand side, we can see a geometa moth, which again observed at Mushrif Park. Following the same methodology, When was this seen? November 2024. And this species is expected in winter in the UAE, where it was in shade near some fresh water and in green shrubs in the park and actively avoiding the sun, flitting around at dusk. And here again, so from experience, I would guess that this is a geometer moth. But to place in its genus, again, I would need to rely on texts and on examination of specimens online. So two telltale points here. Point one is that this is a geometer moth shaped like one and it certainly belongs to one, and it leads me from on the right track from experience. Second is that the serrated antennae indicate that this is a male, and consultation with experts indicates that this is possibly Esturgia parviacea moth. A couple of more case studies, which I'll circle back to if we have time. But this illustrates how we can go down rabbit holes while attempting identification of arthropods. Coming to the next one, basic odonate identification. So this is again the body parts of dragonflies and damselflies, sourced from azdragonfly.org. Some specific salient features which I would like to emphasize. Wings at rest. So damselflies tend to have their wings folded close to their body. Forewing versus hind wing. There's a difference in size in dragonflies compared to damselflies. Eyes separation, again, a very easy way to identify whether what you're looking at is a damselfly or a dragonfly. Flight pattern, most damselflies tend to have weak flight compared to dragonflies. And lastly, size. Damselflies tend to be smaller in general than dragonflies, but there are large species of damselflies as well. So this is as far as the species go. Coming to the next one, the differences between dragonflies and damselflies go. And coming to the next slide on odonate identification. So again, on what I have here is a photograph taken via mobile of a female vagrant emperor, Annex epiphyre, seen in Audmeta in Dubai, near an urban park. When was this seen? November 2024. And this species again is expected in winter in the UAE. Distinctive visual cues, a blue saddle on the second abdominal segment, and second you can see the appendage at the end of this particular dragonfly, which indicate this is a female. So the appendages are separated. This doesn't have a full broodling like Annex parthenope, which is a similar looking species. On the left hand side, you will see the description which is available on British dragonflies, because this dragonfly is seen in many parts of the world, including Britain. So this is basically 3 case studies on identification. I'll quickly go to the case studies from which indicate the not too easy job of identification of arthropods even for experts. So this is a moth called Gandhara typica. This page didn't exist on Moths of India, and I had a chance to contribute the recent photographic records of this. This is the first entry on a moderated website. And these are this is a thread on iNaturalist, which indicates how the process went about to identify this particular species. So we had initially placed it in the genus Churinga, and consultation with experts. This was actually a rabbit hole which led us down to completely wrong track, and none of the references were actually matching what we had seen in the field. After several back and forth and a lot of homework done by everybody, and a lot of deliberation on the thread, we finally arrived at this particular species of moth, for which a fresh page also needed to be created on iNaturalist. So this is just an illustration to show that experts are also human beings, and the process of arthropod identification is revelatory for all. Now coming to resources, so books and magazines you can see, which some of which I've mentioned and some of which you may be aware of. The Emirates and Natural History is a good place to start. Old issues of Tribulus and Gazelle also are rich records of at least knowing what species have been observed in the UAE and Oman. The book Arthropod Fauna of the UAE is something which I have not yet got the hard copy of or the soft copy. I'm hopeful to read it one day. And the Insect Crisis, of course, is more on the motivation side. If you want to get a motivation as to why to study arthropod, this is an eminently readable book released in 2020. Online resources shared on the right hand side. So again, I would encourage, especially for arthropod identification, Biodiversity Library is a great resource which you can search by species named to see the scanned copies of fairly old textbooks and specimens which are available from natural history museums, and the remaining ones have been referenced on the slides. So this is basically it from my end. Thanks a lot for the opportunity to present to the DNHG.